Hello, students, and welcome to another session for Basics in Biology NPTEL. I see there is a thin population today, but it's all fine. It's already 3:05 p.m. We will start the class, and I'll keep admitting other students who are requesting to join this call. Meanwhile, some basic set of instructions for all of you as well as anyone who will be watching this video. So, this session will be a live interactive session, but I would request all the participants to turn off their video chat, turn off their video call and their mics during the whole session. You can interact with me by posting your queries and comments in the chat box. I'll be constantly looking at the chat box for your responses. This will be a two-way interactive session in which we will try to solve an example assignment related to the lectures of the particular week. So today we'll be solving lecture week 4. So all the pertinent lectures in the week 4 module will be covered, and similar to the quizzes or assignments that you receive in your actual course, this will be an example assignment which will help you understand or clear your doubts and attempt the real quizzes and questions as well. Right? So with that of a basic introduction, let me just share my screen and present my slides. Entire screen. I got to know that so all these videos are recorded. This class is a recorded class, as well as the slides will be provided to you via the NPTEL. I just realized that the videos that I have been recording only covers one corner of the whole class or presentation. I'll try to mitigate that in my laptop or computer to upload a full screen video. Okay, I'll just share my screen and present my slides. Yes. So can everyone see my slides? Is everyone able to follow? I'll just put it on the presentation mode. You can just let me know in the chat. Yeah, great. So my name is Ashutosh Behra and I am a teaching assistant for this course Basics of Biology NOC 25 BT02. The course instructor is Professor Vishal Trivedi at IIT Guwahati. So I will be helping. I am a PhD and a PMRF scholar who is helping NPTEL take these live classes. So my job is to help the students as well as anyone who is attending these live classes for solving an example assignment and to clear their doubts. So what I have prepared in here in this video as well is a interactive presentation in which I have 15 questions very similar to what you will face in the actual quiz, and then I have extra slides worth of information trying to cover the basics of these questions so that at least when you attend these live classes or you watch these videos, then for these questions and concepts related to these questions you will be clear of. Okay, as I have already said, I'll keep admitting any student who requests to join in this link while we'll begin the class. So the first question of this class is on the screen. Which of the following is a combination of enantiomer? This is the first question. Which of the following is a combination of enantiomer? Option A, L-glucose and D-glucose. Option B, D-mannose and D-glucose. Option C, D-glucose and D-galactose. Option D, D-mannose and D-galactose. So there are four options here. I would request all the participants to participate in this question answer. You can post whatever you think your answer is in the chat box. I'll be following the chat box in a minute. Let us give some time for everyone to post their answers, as well as once I've collected all the answers, I'll reveal the correct answer to this question. Which of the following is a combination of enantiomer? I'll wait for a minute and collect some answers. I see there are some answers in the chat box. Ramya has answered option A. Aparna, option A. Aishwarya is also saying option A. Okay, the answers are well received. Some time has passed. Now is the time to reveal the correct answer. Let me just switch on my pointer. So, which of the following is a combination of enantiomer? And the correct answer is option A, L-glucose and D-glucose. Very well done, Aishwarya, Ramya and Aparna. You have answered very correctly. So let us understand what is an enantiomer when we pose this question. So what are enantiomers? Enantiomers are nothing but a pair of compounds. So the enantiomers are always two compounds with exactly the same connectivity but opposite three-dimensional spaces, right? Enantiomers are not the same as each other. One enantiomer cannot be superimposed on the other. Enantiomers are mirror images of each other. So when we think of a molecule, you first have to write it in a Fischer projection or in the 3D plane. If you understand, how does a molecule arrange itself in a three-dimensional world, right? For an example, for understanding enantiomers, we have glyceraldehyde. The same molecule can have two enantiomers. One is the D enantiomer and the other is an L enantiomer. So this is one particular type of three-dimensional arrangement of the molecule where the C double bond O carbon, this carbon which is COH and CH2OH, all these three are arranged in a particular plane which is exactly the mirror image, non-superimposable mirror images of L-glyceraldehyde. Thereby we have two enantiomers of glyceraldehyde, the L and the D enantiomers. Now what are these L and D enantiomers? L and D enantiomers refers to two mirror image from a chiral molecule. So these molecules are called chiral, which means this carbon is a chiral carbon, the carbon that we see here, where I'm pointing to at, because this carbon is connected via four bonds to four different functional groups. One is just a hydrogen, here is a C double bond OH, this is OH group and this is CH2OH. Now when you have four unique groups attached to a single carbon, that kind of a carbon is called a chiral carbon. Now in a chiral carbon or in a chiral molecule, in a molecule which has such carbons or any elements, there exist enantiomers which can refer to two mirror image forms of the same chiral molecule. And why are we even studying enantiomers? Enantiomers are prevalent in amino acids, sugars, and they are non-superimposable like left and the right hands, right? They are not superimposable. Now what is chirality again? A molecule is chiral if it has asymmetric carbon atom attached to four different groups. So this is what I was explaining just a minute ago. What molecule would be a chiral molecule? If it has an asymmetric carbon with four different groups. Now what does this L and D mean, where the option was L and D glucose? L stands for levo and D stands for dextro. So the designations L and D come from Latin for the left and the right, when we say the left and the right hand. They are based on the molecule's similarity to the reference compound glyceraldehyde. So most of the times for checking the chirality or administering whether the enantiomer is an L enantiomer or a D enantiomer, we often use the reference molecule as glyceraldehyde. The three carbon compound, right? This is the structure here. This is mostly used as a reference for finding out what enantiomer this class belongs to. Okay. And what is the biological relevance of studying enantiomers, or where do we find that? Do we even find it in the real life? The answer is yes. So this is a structure of D-glucose and L-glucose. Now if you superimpose the structure of glyceraldehyde here, you can see the same CH2OH and this can be treated as the R group with a CO double bond OH and the position of OH group, which makes it an alcohol. This alcohol group, the OH group, is towards the right side here. That is why it is a D glucose or the dextro glucose. And on the left side, it is the L glucose, because these atoms arrange differently when viewed in a three-dimensional system. And amino acids, naturally occurring amino acids in proteins, are mostly always in the L form. Okay, you have to remember these two pieces of information. Amino acids are generally in the L conformation or L enantiomers, whereas sugars, naturally occurring sugars, are typically in the D form. So wherever now when you encounter such compounds like sugars or amino acids or proteins, you now know that sugars are naturally the dextro type and amino acids are naturally the levo type, right? And L and D of a same amino acid or a same sugar are non-superimposable mirror images of each other, which are chiral molecules. So these enantiomers do have biological relevance, and as students have correctly answered, the answer to this question was L glucose and D glucose are the correct enantiomers. Is it clear to the students? Just let me know in the chat box, then I can move to the next question. Very good. Thank you. Okay. Moving on to the next question. In glycolysis, which of the following enzyme helps in conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate? This is the question. In glycolysis, so we are talking about glycolysis, which of the following enzymes helps in conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate? Option A, phosphohexose isomerase. Option B, phosphotriose isomerase. Option C, phosphofructokinase. Option D, phosphoglycerate kinase. These are some of the enzymes which may or may not be in glycolysis, but you have to find out the enzyme that is involved in this particular step, dihydroxyacetone phosphate to glyceraldehyde 3-phosphate conversion. Let me wait for a minute to collect some answers, as well as for the people who will watch this video, and then I'll check the comment box. I'll encourage all students to at least participate in the class. Even if your answer is wrong, you do not have to shy away. I will be revealing the correct answer, as well as we will understand the concept behind why the correct answer is the correct answer, right? So please participate. You just have to post your options in the chat box. Think of the question for a while and try to reach the correct answer. Come on, students, which option do you think is the correct answer? Glyceraldehyde 3-phosphate, does it clink a bell? What is the step in which a glyceraldehyde 3-phosphate is involved? And then there is a conversion of dihydroxyacetone phosphate to this molecule, glyceraldehyde 3-phosphate. If you remember the whole pathway of glycolysis, then this will be easy. Okay, I see some answers in the chat box. Very good. Good marks, all marks for trying. Let us see whether your answer is the correct or not. Aishwarya says B. Ramya sir, maybe B. Or Aparna and Aarti and Isha are all saying B. I would like to tell you guys that you guys are absolutely correct. Well done. Your memory is really good. Correct answer for this question is option B, phosphotriose isomerase. So let us understand, let us look at the glycolysis pathway in detail and what are these two compounds and where does this conversion happen and which is the enzyme that plays the role. Okay, so on my left is a big pathway of glycolysis which many students struggle to remember, but once you go through it 2 or 3 times you can observe some similar patterns, right? In glycolysis we start with a molecule of glucose, single molecule of glucose. Now glucose is phosphorylated. And where does this phosphate come from? This phosphate comes from the molecule ATP, which is the energy currency, right? Now, ATP is not just the energy currency, it is also a good phosphate donor. So, what does happen? The phosphate from ATP is transferred onto the glucose, now generating glucose 6 phosphate. And what is the enzyme that does this? This enzyme is called glucokinase or hexokinase, since the glucose is a hexo 6 membered ring, right? So glucose to glucose 6 phosphate. Then comes an enzyme phosphohexose isomerase. Now what does this do? Now whenever there is an enzyme which is an isomerase, isomerase means rearrangement of the carbons to form a new compound. It will rearrange the molecules, atoms. And whenever there is kinase, kinase is an enzyme which will add phosphate group to its target. Now it is a glucokinase, so it will add phosphate group to glucose. That is why it is called glucokinase. So it will make glucose to glucose 6 phosphate. Now we have phosphohexose isomerase. So it will rearrange this molecules present in the glucose 6 phosphate to form fructose 6 phosphate. Now fructose is a 5 membered ring, right? So once we have fructose 6 phosphate, then there is another kinase, phosphofructokinase 1. So now this is phosphofructose, like, right? Fructose 6 phosphate or phosphofructose. The enzyme that now catalyzes its double phosphorylation is phosphofructokinase. Now there are two phosphate groups added onto the fructose which was rearranged from the glucose. So now we get a product fructose 1,6 bisphosphate, right? Now this is an important step here. From fructose 1,6 bisphosphate there is breakdown of this 6 membered carbon ring into two molecules of phosphoglyceraldehyde. Now this is one of the compounds mentioned in the question, and this is done by aldolase or through the other pathway where fructose 1,6 bisphosphate is converted to dihydroxyacetone phosphate. Now dihydroxyacetone phosphate, upon the action of the enzyme triose phosphate isomerase or phosphotriose isomerase, converts it into 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate, right? So this is the enzyme of our interest. Now if you look at this reaction closely, this is also an isomerization reaction. Now when I say isomerization, the number of carbons will remain the same, but the arrangement will change. So this is an isomerization reaction carried out by triose phosphate isomerase, right? So we have dihydroxyacetone phosphate, which looks somewhat like this. A carbon here, here. So it's a 3-carbon molecule with a phosphate and a ketone molecule. So dihydroxyacetone is a ketone. But then from the ketone we generate an aldehyde. So this kind of isomerization is called ketone to aldehyde, or this ketone will now rearrange its atom to form aldehyde. So then we form glyceraldehyde 3-phosphate by this isomerization reaction, which is catalyzed by phosphotriose isomerase. Now DHAP, which is the short form of dihydroxyacetone phosphate, is a ketose, contains a ketone group. G3P, which is glyceraldehyde 3-phosphate, is an aldose, contains an aldehyde group. This is a reversible reaction, but under glycolytic conditions, it favors the formation of glyceraldehyde 3-phosphate because G3P is quickly used in the next steps. So this isomerization reaction is a reversible reaction. Glyceraldehyde 3-phosphate can also form dihydroxyacetone phosphate. It can turn back the rate of reaction, but it never happens such in a glycolysis pathway because Glyceraldehyde 3 phosphate, as soon as it is produced, is consumed in the further reactions via these enzymes, right? This is not the end product via an intermediate, rather an intermediate which will be fed into the glycolysis pathway to generate energy and pyruvate, right? So since this product is taken away from the equilibrium, taken away from the equation, equilibrium shifts always towards the glyceraldehyde 3 phosphate formation, right? Sorry. So we briefly understood some steps of the glycolysis leading to our conversion enzyme, the triose phosphate isomerase or phosphotriose isomerase, which carries out an isomerization reaction between a ketone, which is a dihydroxyacetone phosphate, to a glyceraldehyde 3, to an aldehyde, which is glyceraldehyde 3 phosphate. Is it clear to the students? I hope with this you can understand why we say this is an isomerization reaction and how to identify enzymes, because glycolysis has lots of enzymes. It's always very difficult to remember the enzyme names, but once you understand the concept behind what is the property of an enzyme and what kind of reaction it catalyzes, it will be easier for you to understand. Okay, cool. Moving on to the third question. Third question says the DNA or RNA are acidic in nature because of its dash. Option A, nitrogen base. Option B, sugar molecule. Option C, phosphate group. Or option D, both nitrogen and sugar molecule. The DNA or RNA or nucleic acid in general, why they are known as nucleic acid? Why acid? Why are they acidic in nature? Now there are some components or features of the nucleic acid mentioned in the options. Which of these components do you think imparts acidity to the nucleic acid? Is it nitrogen base? Is it a sugar molecule? Is it the phosphate group? Or is it both the nitrogen and the sugar molecule? Let me know in the comment box what do you think is the correct answer. I'll wait for a minute for everyone and then I'll check the chat box. This is a relatively easier question for you guys, so I have no doubts that you will answer it very correctly. I'm sorry. Please excuse me. The nucleic acids, right? The DNA or RNA. What makes it acid? What makes it an acid? Basic definition of an acid or a base or the properties, right? Okay, I see some answers in the comment box. Aishwarya C, Aparna is saying C, Ramya is saying C, and Arati has also given explanation. Very good. C because of negative charges and release of protons in solution. So let me tell you that C is the correct answer. Well done, guys. I think we have a brilliant bunch today. We are almost hitting all correct answers, right? So the phosphate group present in the nucleic acid. Let us understand some of the all these components present in the nucleic acid. So here is a structure of both of an RNA and a DNA. RNA, this is a single stranded RNA. We have all these bases. C for cytosine, the blue ones, green guanine, adenine, A, and uracil. These are the four bases, nitrogenous bases or nucleobases of RNA. And this is the RNA, how it looks like in a helical arrangement. These are the nucleobases. And this backbone, this greenish gray line, is a helix of sugar phosphates. So this backbone is formed by sugar and phosphates. and this RNA stands for ribonucleic acid. And then this is the structure of a double helix DNA. In DNA, we all have all the components, the cytosine, guanine, adenine, and all these three components are same in the DNA, but only we have a replacement of uracil with the thymine. This occurs in the DNA. Now, DNA is a double stranded. The base pairs are like this with the backbone forming of the sugar and phosphate. Now, out of all these are all the components that we saw in the options: the sugar phosphate backbone, the nitrogenous base, the phosphate, or both the nitrogen and the sugar. Now, let us understand what brings the acidity to nucleic acid. Now, the group that brings the acidity to nucleic acid is the phosphate group. Phosphate group has 3 minus or 3 negative charges, PO4 3 minus. Each nucleotide in nucleic acid, the DNA or RNA, contains a phosphate group attached to the sugar backbone. Now, if you remember the helix that we saw in the previous slide, each molecule of a pentose sugar, the 5-membered ring sugar, for RNA it is ribonucleic acid, and for DNA it is deoxyribonucleic acid. Right? There is a subtle difference which brings enormous changes in the properties of both these nucleic acid, the DNA and the RNA. Now, each such pentose sugar is attached to another pentose sugar via a phosphate backbone. This is the phosphate that links both of them. Right? And these phosphate are attached to one of the carbons in the 5-membered ring. The phosphate group has a negative charge due to its oxygen atoms, and it can release protons in aqueous solutions, giving nucleic acid their acidic properties. As rightly mentioned by Aarti and Aparna as well, the phosphate group,
Thank you, sir.
phosphate group resembles the phosphoric acid H3PO4. So the phosphate group has negative charge and it can release protons when dissolved in the solution. This is why DNA and RNA are called nucleic acid, because they are negatively charged and acidic at physiological pH. So even at the pH of blood and the body, which is 7.4, due to ionization of these phosphate groups, we call them nucleic acids. So here below I have listed all the components present in one nucleotide. This is called, also known as, nucleoside triphosphate. This is the pentose sugar that I was mentioning. This is the CH2OH group to which the phosphate is attached. This is one of the nucleotides. So this is ATP with triphosphated. Such molecules, the NTPs, ATP, GTP, CTP, TTP, and UTP, form one functional and structural unit of the DNA. So we have the sugar here, the pentose sugar linked to the phosphate, and nitrogenous base linked via a glycosidic bond. So this portion is called a nucleoside with only the sugar and the nitrogenous base. This is nucleoside monophosphate, nucleoside diphosphate, and triphosphate. And the whole single nucleotide is with a phosphate linked to the other nucleotide, right? And the nitrogenous bases can be pyrimidines or purines. Pyrimidines are cytosine, thymine, and uracil, while purines are adenine and guanine. Okay, I think it's clear to everyone. So let's just move on to the next question. Question number 4. Which of the following are decarboxylating enzymes in Krebs cycle? I'll read it again for you guys. Which of the following are decarboxylating enzymes in Krebs cycle? Option A, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. Option B, alpha ketoglutarate dehydrogenase, aconitase. Option C, alpha ketoglutarate dehydrogenase, succinate thiokinase. And option D, isocitrate dehydrogenase and aconitase. Which of the following are decarboxylating enzymes in the Krebs cycle? Let me give you a small hint which you already must have remembered. Krebs cycle is nothing but the citric acid cycle or the tricarboxylic acid cycle. And there many steps involved decarboxylation. Now you have to remember what all enzymes are there in the Krebs cycle and which of these enzymes do you think are decarboxylating enzymes. Let me know your answers in the chat box. I'll wait for a minute and then we'll discuss the answers. Okay, it's time to check the comment box. Ramya has answered it's A, Aishwarya A, Aparna A, and there is another comment. Since these enzymes lead to the loss of two CO2 molecules per turn of the cycle. Good. Now let us see what is the correct answer to this question. Now, which of these enzymes are decarboxylating enzymes in the TCA cycle or Krebs cycle? Correct answer to this question is option A. I cannot emphasize enough that this is the correct answer. Well done, guys. Option A, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Now let us understand what is decarboxylation. As correctly mentioned in the chat box, decarboxylation is nothing but removal of carboxyl group or a CO2 molecule from any reaction is decarboxylation. So this is a schematic here showing rCO2 phosphate. This is a molecule. Removal of CO2 and plus H plus forms this compound. This is a decarboxylation reaction on the top. Then on the bottom we have isomerization reaction, in which due to addition of proton, both of them go through this intermediate. Now the P and R groups are interchanged, right? So this is an isomerization, while decarboxylation means removal of CO2. Decarboxylation via decarboxylating enzymes refers to a chemical reaction where a carboxyl group is removed from a molecule, releasing carbon dioxide. So many decarboxylation reactions lead to release of carbon dioxide. Now this is a big complex picture of the tricarboxylic acid cycle or citric acid cycle or Krebs cycle after the scientist who has discovered it. Let us now move through the steps of the Krebs cycle. So we start off with pyruvate. Then pyruvate gets converted to acetyl coenzyme A. Now pyruvate is the end product of glycolysis, which we encountered in the second question maybe. Pyruvate converts to acetyl coenzyme A. Acetyl coenzyme A now feeds into the tricarboxylic acid cycle, is the starting material. Citrate synthase enzyme which acts here, and formation of citrate, right? And then removal of water by aconitase and forms cis aconitate. Then aconitase acts then again to form isocitrate. So the citrate is now rearranged to form isocitrate. Then with a utilization of NAD plus forming an NADH, isocitrate dehydrogenase catalyzes this reaction, conversion of isocitrate to alpha ketoglutarate via removal of a CO2. So here we have reduction in one carbon number. We form alpha ketoglutarate. Then in the next step, we have the enzyme alpha ketoglutarate dehydrogenase converting alpha ketoglutarate to succinyl coenzyme A. This is also a decarboxylation reaction in which another molecule of CO2 leaves the cycle. So we started off with acetyl coenzyme A, 2 molecules of pyruvate gives a 6 carbon structure, the citrate, and then we have loss of 1 carbon here in a decarboxylation reaction and here as well. So loss of 2 carbons, right? Then we form the 4 carbon compound, the fumarate, malate, oxaloacetate, and then in subsequent steps we generate NADH as the molecule and utilize ATP as well. So this is the legend. But the focus of our question was decarboxylating enzymes, which are these enzymes: isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase, which play an important role in reducing the carbon number from isocitrate to alpha ketoglutarate, and the subsequent step alpha ketoglutarate to succinyl coenzyme. Very good, you guys have answered really well. Is this clear to the students? I know this can be quite complex for a Krebs cycle, but Krebs cycle is inevitable, right? You have to understand Krebs cycle and glycolysis very well. Great. Thank you. Moving on to the next question. Question number 5. TPP, thiamine pyrophosphate, is required in dash step of Krebs cycle. Okay, so this series will follow some of these questions from Krebs cycle, which I just showed in the first just showed in the previous slide. So I think it would not be a difficult question to answer. TPP, if you remember, thiamine pyrophosphate, is required in dash step of Krebs cycle. Option A, decarboxylation of oxalosuccinate. Option B, decarboxylation of alpha ketoglutarate. Option C, decarboxylation of aconitase. Option D, decarboxylation of pyruvate. Where do you think TPP is utilized? Thiamine pyrophosphate. Now you have to remember what kind of a molecule is thiamine pyrophosphate and where could it potentially be employed. I will wait for some time and then check the comment box for answers. I'm sorry. I may have to edit this answer as well. Okay. I see many people have answered in the chat box. Option B, Ramya, Aishwarya and Aparna are all saying option B. Okay, let me reveal the correct answer to this question. TPP is required in dash step of Krebs cycle. The answer is option D, decarboxylation of pyruvate. But I know the option B is also correct. Decarboxylation of alpha ketoglutarate also requires thiamine pyrophosphate. So I'll just change my marking here. The actual answer would be actually option B, because pyruvate technically is not a part of Krebs cycle but is a starting material. Pyruvate is actually the end product of glycolysis, which is then utilized into the Krebs cycle, right? So I'm striking both of these options correct here. Decarboxylation of pyruvate as well as decarboxylation of alpha ketoglutarate. Let us understand how does it do the decarboxylation of pyruvate and what is TPP. So there is a big enzyme complex called pyruvate dehydrogenase complex, and TPP stands for thiamine pyrophosphatase, and this is also called a link reaction. It's a link between, this is a link reaction. Pyruvate converts to acetyl coenzyme A and CO2. So this is a decarboxylation reaction. Two molecules of pyruvate convert into acetyl coenzyme A with one CO2 release. TPP is a coenzyme in the E1 component of the pyruvate dehydrogenase complex. So this is a big enzyme complex, pyruvate dehydrogenase. TPP, the thiamine pyrophosphate, is a coenzyme in the E1 component. So we have various components of this enzyme. This is the enzyme, the whole enzyme complex. It helps decarboxylate pyruvate and transfers hydroxyethyl group to the lipoamide cofactor. So if you see here, this is the pyruvate. Pyruvate is channeled to TPP. TPP holds it. So it is hydroxyethyl TPP. Now this will be transferred into the acetyl lipoamide. This is all the component of the enzyme complex. So oxidized lipoamide will take on to the hydroxyethyl TPP. Now this will now act on coenzyme A, the coenzyme A, and then turn it into acetyl coenzyme A. Now this will be fed into the Krebs cycle. So this is one of the reaction that where TPP is involved, conversion of pyruvate into the acetyl coenzyme A, feeding into the Krebs cycle. Now this reduced lipoamide, which was oxidized, then transferred with the hydroxyethyl, then this group was transferred to coenzyme A. Now this reduced lipoamide can recycle back with the utilization conversion of FAD to FADH2 to transfer back into the oxidized lipoamide. So here TPP is the first coenzyme which receives the carbons present in the pyruvate and transfers it to the acetyl lipoamide, which will now get transferred to the coenzyme A. Similarly, in alpha ketoglutarate decarboxylation also, TPP is the coenzyme. This step links glycolysis to the Krebs cycle, supplying acetyl-CoA for entry into the cycle. So this is something which is above the Krebs cycle, which is feeding into the Krebs cycle. So this is also known as link reaction, conversion of pyruvate to acetyl coenzyme A, so that acetyl coenzyme A can now enter the Krebs cycle. And why is TPP so important? Why does it act as a coenzyme? What are its special properties? It stabilizes carbanion intermediates during decarboxylation reaction. So this is the most important property of thiamine pyrophosphatase. It can stabilize the carbon intermediates that form during decarboxylation reaction. And TPP is something which is formed when you take enough thiamine or vitamin B1. So deficiency in thiamine can impair these steps, leading to issues like beriberi or disrupt energy metabolism. So this is an important component of the vitamin B1 or thiamine complex, forming the coenzyme which is necessary for conversion of pyruvate to acetyl-CoA, as well as in the alpha ketoglutarate step decarboxylation reaction. It acts as a coenzyme. I'm sorry for the mistake in the answer. Your answer was definitely correct, option B, but option D will also be the correct answer, where you have conversion of pyruvate to acetyl coenzyme A. Is it clear to the students? This is the schematic of the Krebs cycle I'm showing, and where TPP can be utilized in these steps as well as in the alpha ketoglutarate. Okay, cool. Moving on to the sixth question. Amino acids such as glutamine, proline, and arginine are formed using dash intermediate from Krebs cycle. This should be an easy answer for you guys. Amino acid such as glutamine, proline, and arginine are formed using dash intermediate from Krebs cycle. Option A, fumarate. Option B, oxalosuccinate. Option C, alpha ketoglutarate. Option D, succinate. Which of these will feed into amino acid biosynthesis pathway such as glutamine, proline and arginine? And one of these is an intermediate in the Krebs cycle. Let me wait for a minute and collect some answers. Come on, guys, you have been answering everything correctly, so well and so good. That's very good. That's very great. I want more correct answers, or at least participation from all the students. Which of these will feed into amino acid synthesis pathways? Is it fumarate, oxalosuccinate, alpha ketoglutarate, or succinate? I just hope you're not googling it meanwhile I'm waiting for answers. We are here to try to understand the concept so that you won't need Google when you try to remember this. Which of these will serve as an intermediate? Is an intermediate from Krebs cycle that can serve or feed into the amino acid metabolism biosynthesis pathway? Okay, let me check the comment section. Ramya and Aishwarya has answers option C, and I don't think there are more answers. Okay, so let me show you what is the correct answer to this question. Which of these intermediates from Krebs cycle can feed into amino acid biosynthesis pathway? The correct answer is as mentioned by Ramya and Aishwarya. It's alpha ketoglutarate. Option C is the correct answer. Well done. Now let us understand what is alpha ketoglutarate and how does it lead to amino acid production, right? So this is a simplified schematic of the TCA cycle showing two important components, the alpha ketoglutarate and oxaloacetate. Now alpha ketoglutarate converts into glutamate. So alpha ketoglutarate to glutamate. Now glutamate is number one, glutamate is an amino acid itself, and glutamate can act as a precursor. Glutamate can act as a precursor to other amino acids such as glutamine, arginine, proline, and glutamine can then form histidine. Now another intermediate, oxaloacetate, the 4-carbon compound, can form aspartate. Then aspartate can feed into pathways for synthesis of lysine, threonine, methionine, cysteine, serine. So according to our question, amino acids like glutamine, arginine and proline come through the intermediate glutamate, which is formed from alpha ketoglutarate, one of the intermediates in the TCA cycle. While oxaloacetate may lead to aspartate, lysine, threonine, methionine, cysteine and serine production, which can also lead to glycine production. Now how does this reaction happen? This is the schematic here. Alpha ketoglutarate, this is the molecule, undergoes amination reaction. Amination means addition of an ammonium group, NH4, with utilization of an NADPH to NADP, glutamate dehydrogenase. This is catalyzed by the enzyme glutamate dehydrogenase, forming glutamate. Glutamate is an amino acid itself, so the NH3 group here attaches to this carbon, alpha carbon, and then glutamate can serve as a precursor acted upon by glutamine synthetase to form glutamine, addition of another ammonium group, right? So this is how alpha ketoglutarate can feed into amino acid synthesis. The Krebs cycle, citric acid or TCA cycle, plays a critical role in synthesizing several non-essential amino acids by providing key intermediates that serve as precursor for amino acid biosynthesis, as we just saw, alpha ketoglutarate to glutamate. Here's how important intermediates are connected to amino acid synthesis: alpha ketoglutarate to glutamate, and then glutamate can give rise to other amino acids. Alpha ketoglutarate is a central intermediate that undergoes transamination to form glutamate. So this reaction is called a transamination reaction, where an amino group is added to the alpha carbon in the alpha ketoglutarate. Alpha ketoglutarate plus NH3 plus NADPH gives glutamate plus NADP plus H2O. This is what I was showing here. Alpha ketoglutarate plus NH3 plus NADPH gives glutamate catalyzed by glutamate dehydrogenase. Now glutamate is a precursor for glutamine via glutamine synthetase, proline via cyclization and reduction of glutamate, arginine via the urea cycle starting from glutamate and glutamine. Is it clear to the students? So those who have answered correctly, well done. And those who could not answer, this is the correct answer, and this is the concept behind how alpha ketoglutarate can feed into the pathway of amino acid biosynthesis. Great. I have received your response. Thank you. Now let's look at the seventh question. Which of the following molecule has maximum number of carbons in their structure? Okay, now moving on to the fatty acid and lipids part. Which of the following molecule has maximum number of carbons in their structure? Option A, arachidic acid. Option B, stearic acid. Option C, palmitic acid. Option D, myristic acid. Which do you think has maximum number of carbons in their structure? I'll wait for a minute and collect some answers. Okay, it's a minute now and I see some answers. Ramya says D, myristic acid. Aparna is also going with D, whereas Aishwarya is going with A. Oh, Ramya has changed her answer to stearic acid. Okay, I see there are multiple answers coming up. So this question might need some clarification. Okay, let me reveal what is the correct answer for this question. Correct answer to this question is, which of these acid has maximum number of carbons? Correct answer is option A, arachidic acid. So only Aishwarya has answered it correct. Others got confused a little bit. It's okay. It's absolutely fine, right? There are so many acids. There are many number of carbons. All these look very similar. Let us look at all the structures of these acids. So where is our star player, which is the arachidic acid? So this is arachidic acid. Arachidic acid has how many carbons? Let us count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 carbons. And saturated acid as well. So there are no double bonds. Double bonds means the acid is unsaturated and the degree of unsaturation. So this is a saturated acid, fatty acid, contains 20 carbon atoms. Myristic acid on the other hand, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 carbons for myristic acid. For palmitic acid, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 carbons for palmitic acid. And we have stearic acid, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So we have acids. All the options were ranging from 14 carbons to 16 carbons to 18 carbons, and finally we have 20 carbons. So arachidic acid is a saturated fatty acid with 20 carbons. So that is the correct answer. Maximum number of carbons among all these fatty acids. There are other acids that I have mentioned here in the structures for you guys to remember. Is it clear to the students? Well, this was just a memory based question, so it's okay if your answer was not correct. But congrats to the one who answered the correctly. A short question with a short answer. Is it clear to the students? Okay. Great. Moving to the next question. Glycerol is a dash carbon molecule. Come on, this is the easiest possible question. Glycerol is a dash carbon molecule. Option A, 2 carbon molecule. Option B, 3 carbon molecule. Option C, 4 carbon molecule. Option D, 5 carbon molecule. Glycerol. How many carbons are there in glycerol? I'll wait for a minute for all those who will also be watching this video, and all the students who are present in the live class. Please let me know what is your answers. Take a minute, think about it. How many carbons do you think a glycerol molecule has? Glycerol, guys, glycerol, come on. Glycerol is the simplest molecule, forms the backbone of lipids, right? If you remember a single lipid, then you can know how many carbons does glycerol have. Okay, it's time for me to tell you what is the correct answer, but I can see in the comment box Aishwarya is saying B. Ramya was first confused A or B, but then she is at B. Aparna says B, 3 carbon. Well, I cannot deny you guys that you guys are correct. Option B is the correct answer. Glycerol is a 3 carbon molecule. So here on my right is a structure of glycerol. 3 carbons linked to each other. Now carbon has 4 valencies, right? So 2 of them are hydrogens, 1 oxygen. 1 hydrogen, 1 oxygen. and the last one, two of them hydrogen and oxygen. Now, if you remember the earlier questions where we talked about enantiomers, glyceraldehyde is the aldehyde of glycerol. So this is the alcohol, we have glyceraldehyde, and which is the aldehyde of this alcohol. That also contains three molecules. So from there we can, if we just remember, we can easily recall that the OH group present on the right or the left in reference to glyceraldehyde terms the L and D enantiomers. So from there it was easy to guess that glycerol is a 3 carbon molecule. Now what I am showing here is the formation of a triacyl glycerol, which is a single structural and functional unit of a lipid. Glycerol reacts with fatty acid. So this is a long chain fatty acid. The fatty acid that we encountered in the last question was arachidic acid. Let's say this is arachidic acid or any other long chain fatty acid. This has the acidic COOH carboxylic group here. This will react with the oxygen with the removal of a water, forming this ester linkage, forming the triacyl glycerol. See, each molecule of the each carbon now replaces its OH group with a bond with the COOH from the acid of the glycerol, forming the triacyl glycerol. Now this triacyl glycerol is a unit of lipid membrane that we see, the plasma membrane. Of course, glycerol is a 3 carbon molecule and has important functions. Triglycerides, energy storage molecules or fat that we call as, and phospholipids, essential for cell membrane structure. So the plasma membrane, the cell membranes, the organelle membranes are all made up of phospholipids. The lipids are made up of this molecule, glycerol and fatty acids, and then we have other amino acids. So it was not a difficult question to answer, and I hope that it's clear to everyone that a glycerol is a 3 carbon molecule with 3 OH groups, which makes it an alcohol. I hope it's clear to everyone. Cool. Very good. Moving on to the ninth question. Nitroglycerin is used as a dash. Option A, anti-cancer drug. Option B, antihistamine. Option C, vasodilator. Option D, lubricant. Nitroglycerin is used as which of these? We are moving really fast. We have a lot of time on our hands. We have reached the ninth question and we still have around 50 minutes of time. So we'll go slow. I'll try to engage everyone present in the class as well as watching the video for answering all these questions. So we are on the ninth question. I've prepared a total of 15 questions for you guys. Now think about it. What could be nitroglycerin's use for humans? Is it an anti-cancer drug? Is it an antihistamine or a vasodilator or a lubricant? I'll wait for a minute and collect some answers. Meanwhile, I'll encourage all the students to participate. We have almost covered 50% of the questions that I had prepared for you guys. So please participate in the other half of them. Nitroglycerin. Let me tell you, nitroglycerin is made with the starting material as glycerol because it has the same glycerol backbone with a 3 carbon. So nitroglycerin, the precursor molecule is glycerol. We are having kind of related questions. In the previous question we had how many carbons does a glycerol molecule have? Now we are talking about nitroglycerin. What is it famously used for? Okay, let me take a look at the chat box. Time is up. Aishwarya says C, maybe C. Aparna at first said C, but now she is also confused. Option A or C. And there are no more answers. Okay, so let us see what is the correct answer for this question. Nitroglycerin is used as a vasodilator. Option C is the correct answer. You guys have both answered well. Aishwarya and both Aparna. Option C is the correct answer. Nitroglycerin is a vasodilator. Now, what is vasodilation? Vasodilation is the widening of blood vessels due to relaxation of the smooth muscle in the vessel walls, primarily in the arteries and arterioles. So, vasodilation means dilation of the blood vessels. So, if you can see my hands, if this is the lumen of the blood vessels. So, when I say blood vessel, it looks something like this. Is an arteriole, normal blood vessel. The empty space in the blood vessel through which active volume of blood flows is called the lumen or the fluid filled space. And then we have the artery walls. Now these artery walls are made up of smooth muscles or involuntary muscles. This is controlled by involuntary center which is present in the medulla oblongata. So that is why our cardiac blood pressure is determined by the thickness of the artery. If the artery is dilated, means it is expanded, the lumen has expanded, then the blood pressure will decrease. If the artery is constricted, then the same flow will happen at a higher pressure, therefore increasing the blood pressure. So in a normal blood vessel, we have normal lumen. This is the wall of the artery. There are RBCs flowing through this. Then we have a vasodilated blood vessel. In a vasodilated blood vessel, widening of the lumen occurs. So there is now more blood flowing with a lesser pressure. So lumen to become larger and there is decrease in pressure. And what happens in vasoconstriction? Constriction means lessening of the volume of the blood, lessening of the artery width, narrowing of the blood vessel, lumen to become narrower and the artery walls become thicker. So vasodilation is expansion of the lumen or bigger lumen, low blood pressure, and this happens due to relaxation of the smooth muscle in the vessel walls. So the vessel walls which have smooth muscles relax, causing this vasodilation. This results in an increase in blood flow and decrease in blood pressure. This is what I was saying. Now blood flow will increase and the pressure will decrease. Now where does nitroglycerin come into picture? So this is a smooth muscle cell on the left and the right as well, showing how relaxation in the smooth muscle cell can be induced via nitric oxide. So compounds like sodium nitroprusside or nitroglycerin or organic nitrates, which can all act as a source for this gas or a compound called nitric oxide. Nitric oxide can then go inside the smooth muscle cells and activate GC. GC is GCAMP, guanine cyclase, AMP guanyl cyclase. Guanyl cyclase is an enzyme which catalyzes the conversion of GTP, guanine triphosphate, to cGMP or cyclic GMP. Right? Now, when there is cyclic GMP excess in the smooth muscle, this leads to relaxation of the smooth muscle. So, whenever there is increase in nitric oxide due to nitroglycerin or nitroprusside or organic nitrates, this NO as a gas can penetrate the smooth muscles. Now in the smooth muscle, NO or nitric oxide activates guanyl cyclase. Now guanyl cyclase can convert GTP into cyclic GMP, which then signals for relaxation. So this is how nitroglycerin can act as a vasodilator. Now when the smooth muscle will relax, it will lead to increase in the lumen, increased blood flow with decreased blood pressure. Nitroglycerin does not directly act on smooth muscle but releases nitric oxide. So this is the key point. It does not directly bind to the smooth muscle but releases nitric oxide, which triggers a biochemical pathway leading to muscle relaxation, which is this: activation of guanyl cyclase, conversion of GTP to cGMP or cyclic GMP, guanine monophosphate, which aids in relaxation of the smooth muscle. and causes muscle relaxation and vasodilation. So this is how nitroglycerin functions as a vasodilator. Is it clear to everyone? Very good. Good. Great. Moving on to the 10th question. An example of docosanoic acid is dashed. What is an example of a docosanoic acid? Option A, behanic acid. Option B, arachidic acid. Option C, caprylic acid. Option D, lauric acid. A docosanoic acid. These are all fatty acids. You have to tell me which of these is an example of a docosanoic acid. For that, you have to know what is docosanoic acid. Let's wait for a minute and collect some answers. Sorry. Okay, let me take a look at the chat box and see what answers have been posted. Ramya says maybe A but not sure. Aparna is saying D, and I don't see anyone else answering. Okay, let me reveal time to reveal the correct answer to this question. What is an example of a docosanoic acid? Option A is the correct answer. Behanic acid is the correct answer to this question. Ramya has answered correctly. Behanic acid is an example of a docosanoic acid. This is the structure of behanic acid. Docosanoic acid is also, so the other name of behanic acid is docosanoic acid, is a long chain fatty acid that is saturated and straight changed. So how many carbons? 22 carbons. It has the chemical formula C22H44O2 and an average mass of 340.592. So these many grams forms 1 molar of this compound. 1 mole, right? And it has 22 long carbons, so it is very very long. So in the other question, we dealt with arachidic acid, which was 20 carbons long, saturated long chain fatty acid. So in a docosanoic acid, doco means 2 more than icosanoic. Now an icosanoic is a 20 carbon molecule, so is docosanoid is 22 carbon molecule. It is the other name of behanic acid. This is just a memory based question, so I think it's clear for everyone. There is a question in the chat box. I think, let me read the question. Okay. Aparna is asking, sir, docosanoic and docosanoic acid are different acids. Is it? Docosanoic. What is this? Docosanoic acid. So I only know docosanoic and sanoic. So san an and ene refers to a double bond. So docosanoic acid is a unsaturated fatty acid. It will have a alkene bond, while a straight chain saturated fatty acid is the docosanoic acid. So every acid, so if you remember the basic difference between alkane, alkene and alkyne, so where carbons are connected through a single bond or a double bond or a triple bond. So single bond are alkane, so this is a docosanoic acid, and in senoic acid there is a double bonded carbon. I hope it's clear to the question. Just let me move back to the video call. Okay, great. Moving on to the next question. Which of the following phospholipid is related to removal of apoptotic cells by phagocytes? Okay, coming on to lipids now. Which of the following phospholipid is related to removal of apoptotic cells by phagocytes? Option A, phosphatidylcholine. Option B, phosphatidylserine. Option C, phosphatidylethanolamine. Option D, phosphatidylglycerol. One of these lipids is related in apoptotic pathway and removal by phagocytes. Before even so I'll take a minute, wait a minute for to collect some answers. I'll encourage all of the participants to please post your answer. We are reaching the 11th question. Soon we'll finish this session. So I don't want anyone to be left out. You should all have a chance in answering these questions, right? You just have to post your answer in the comment box, and even for all those watching in the video, try to guess the answer. Which of the following phospholipid is related to removal of apoptotic cells by phagocytes? Okay, let me have a look at the chat box. Who, what is the answer that have been posted? Aparna and Aishwarya is saying maybe B. Okay. What about others? Are others too shy to post their answers? There are two more students, I see. Mrs. Babu and Ramya. Not sure. Okay. Okay, Ramya. It's fine. It's fine. Okay, so let me reveal what is the correct answer to this question. Correct answer is B, phosphatidylserine. Well done to all those who have answered correct. Phosphatidylserine is such a phospholipid which is very important in physiology. It is related to removal of apoptotic cells by phagocytes. Now, what are phagocytes? Can anyone tell me what are phagocytes? You can just type in the chat box. I hope you know what are phagocytes. I'll keep following the chat box. Can any of the students tell me what are phagocytes? Basically WBCs. Yeah, you're correct. Correct, Ramya. Aparna has given a great description as well. Yes, in that description we can see that the importance of phagocytes. The important term for phagocyte is engulfment. Cells that have crucial role in immune system. Very good. All good answers, all correct answers. Now phagocytes have the special property of eating other cells. Phago means eating and cytes means cells. Cells which eat, which can eat other cells. Now, phagocytes have a major role to clear apoptotic cells. Apoptotic cells means cells which are suiciding or cell death. Whenever there is cell death, to remove these such dying cells, phagocytes are the one which come into picture, which eat those dying cells and clear them off, so that their carbon, their lipids, their proteins and amino acids can all be recycled and used by the body again. Now, in this whole process, phosphatidylserine has a major role to play. We will look at it. So before that, let me tell you what is phosphatidylserine. So this is a lipid chain. This is a schematic of a lipid, any general lipid. We have the glycerol backbone, 3-carbon backbone, then COOR. This is the fatty acid R1, fatty acid R2, and the X group, which now changes the nature of the lipid. This will form the lipid head, and all these fatty acid, long chain fatty acid, will form the lipid tail. Now, phosphatidylserine, which is our candidate here, has, instead of X, we have the serine amino acid attached to the glycerol backbone here, and R1 and R2 can be any long chain fatty acid. When this X group is replaced by choline or ethanolamine or glycerol, it gives all these other options that we saw. Phosphatidylcholine, ethanolamine, phosphatidylglycerol. All of these have distinct roles. But how does phosphatidylserine play a role in phagocytosis? Phosphatidylserine plays a crucial role in apoptosis by acting as an eat me signal on the surface of dying cells, which allows phagocytes to recognize and engulf them during the process of phagocytosis. So exposure of phosphatidylserine on the outer membrane of the cell is an eat me signal. The cell is trying to tell or communicate that, phagocyte, you have to come and eat me or clear me because I am undergoing cell death or apoptosis. So exposure of phosphatidylserine from the inner leaflet of the plasma membrane to the outer leaflet, like this. There are many proteins which are called lipid transporter or scramblase proteins as such, or enzymes, which facilitate the movement of lipids inside and outside. Or, since the plasma membrane is a double membraned structure, from the inner leaflet which faces the cytoplasm to the outer leaflet which faces the extracellular matrix. Now, when the phosphatidylserine is exposing and facing the extracellular matrix, this is an eat me signal to the cell, that a phagocyte can now come, recognize the signal and bind to that cell and engulf the cell during the process of phagocytosis. When a cell undergoes apoptosis, it flips phosphatidylserine to the outer membrane, signaling the nearby phagocytes that the cell needs to be removed. So this is what I was talking about. When the cell is undergoing apoptosis, it deliberately flips the phosphatidylserine from the inner leaflet to the outer leaflet. Now the phosphatidylserine which is exposed will be recognized by the phagocyte by a direct recognition. So phagocytes have certain receptors and bridging molecule which help recognize the phosphatidylserine on the apoptotic cell, and the phagocyte can now engulf the whole cell which is displaying these eat me signals. So phosphatidylserine act as eat me signals in apoptosis, signaling phagocytes for clearance. Is it clear to the students that what is the role of phosphatidylserine in this process of apoptosis? Very good. Okay. Quickly moving on to the 12th question. Sphingomyelin are most abundantly found in dashed. Sphingomyelin. Option A, connective tissues. Option B, bone marrow. Option C, lung epithelial cells. Option D, nerve cells. Sphingomyelin, where is it most abundantly found in the body? Which kind of tissues or cells do you think will be abundant in sphingomyelin? I'll wait for a minute and collect some answers. Okay, it's a minute or so. Let me check the chat box for answers and then I'll reveal the correct answer. Aishwarya is saying maybe D. Aparna says don't know. It's okay. Ramya, sir, it's D since mycelium sheath is key component. Okay. Let me now reveal the correct answer to this question. Correct answer is D, nerve cells. Sphingomyelin is most abundant in nerve cells. Ramya, thank you for that great explanation. You have made my job easier. And Aishwarya, you were correct. It's D. Aparna, it's okay if you do not know. We will know it just in a minute or so. Why sphingomyelin and what is its role in nerve cells? So this is a slide displaying all the kind of sphingolipids that occur in our body and our candidate, which is sphingomyelin. This is a long chain fatty acid and this is the COOH group acid here. We have a phosphoethanolamine group here, and while the other carbon has an amino group attached to the fatty acid, right? We have other members like the sphingosine, ceramides, sphingomyelin, cerebroside, ganglioside. So these are all different because of the sphingosine backbone. This is the sphingosine backbone, right? The NH3 plus COH, C and COOH. So instead of the typical glycerol backbone, we have the sphingosine backbone. Now this OH group can be replaced by other moieties which give it properties. So a sphingomyelin has phosphoethanolamine group attached to it. As explained correctly in the chat box by Ramya, sphingomyelin, as the name stands for, is particularly abundant in neurons, specifically within the myelin sheath that surround neuronal axons. So this is the structure of a neuron. This is the cell body of the neuron, or called cytol. The cyton. This is the axon, cell body, cyton, and the axon. So nerve signals generated here, they transmit through the axon and end up in the axon terminals at synapses, which connect to the dendrites of another neuron. So now the axon of the nerve is covered with a specific kind of sheath called a myelin sheath. Now this myelin sheath is what made up of sphingomyelin. Sphingomyelin makes up this myelin sheath. It is a major component of central nervous system where it plays crucial role in maintaining integrity of nerve fibers and facilitating efficient signal transmission. So this sheath acts as an insulation on the nerves for smooth conduction of electrical signals. So now the electrical signals can transmit very faster and they can be insulated like an insulated wire. They can be insulated and the signal loss will be very less. Now the loss of signal will decrease because of myelinated neurons. That is where the sphingolipids come in play. Sphingomyelin is a major component of myelin sheath. Is it clear to the students? We had a look at the sphingolipids and where does it play a role in. Very good. Thank you. The 13th question is here on display. Dash phospholipid is used as a surfactant. Now, application based questions. Which is the phospholipid that is used as a surfactant? Option A, dipalmitoyl lecithin. Option B, phosphatidylcholine. Option C, phosphatidylinositol. Option D, phosphatidylserine. Which of these phospholipid can be used as a surfactant? Now, in order to answer this, you have to know what is a surfactant. Right? I'll wait for a minute or so and take a look at the chat box where you have posted your answers. Since this is the 13th question, we have two more questions to go and then we'll wrap up this session. So I urge everyone present in this class to participate as well as people who are watching this video. Which phospholipid is used as a surfactant? Okay, let us look at what is the correct answer to this question. Phospholipid, which of these phospholipid is a surfactant? Let me look at the options first. Sorry. Aparna says, I think it's B. Surfactant. Okay, Ramya has a long answer, explanation as well. Surfactant are substances which reduce tension between two compounds, for example, oil and water. Very good, very good. Thank you for the explanation. Time to reveal what is the correct answer. Correct answer to this question is option A, dipalmitoyl lecithin. Option B, phosphatidylcholine, could have been the correct answer, but dipalmitoyl lecithin is a modified phosphatidylcholine which has other functional groups, which is the actual surfactant found inside the body. I will explain in a while why option A is the correct answer. First of all, what are surfactants? As very well explained by Ramya, surfactants are substances that reduce surface tension between two phases. When you mix, for the example, water and oil, they do not mix. Water is hydrophilic while oil is hydrophobic. They have different surface tension, so they do not mix. So if you add surfactant into the mixture, now there are three components. This will help the water and oil to mix. They are also known as surface active agents. How do surfactants work? Surfactants are made of a molecule that are both hydrophobic and hydrophilic parts. So these kind of molecules are also called amphipathic, such as our lipids. When added to a liquid, surfactants reduce surface tension, which increases liquid spreading and wetting properties. Surfactants form molecular clusters called micelles or micelles in a solution like this. This is a micelle. Surfactants can be made from natural or synthetic raw materials. So surfactant is something which will help two different phases to mix like water and oil by forming micelles. Very good explanation. Now looking at what is the surfactant dipalmitoyl lecithin. So dipalmitoyl lecithin is a lungs surfactant. So this is the structure of DPPC. So this has a backbone of a phosphatidylcholine. So phosphatidylcholine backbone, but the palmitoyl groups are added as the fatty acid, and this is a lecithin. Dipalmitol lecithin, or DPPC in short, is found in the extracellular fluid and surrounds the alveoli of the lungs. So in the alveolar compartments there is this DPPC, and what does it do? It decreases the surface tension of the lungs to prevent the lung collapse during breathing. So during breathing, when air and blood are in contact in the alveoli, they form two different phases, and this DPPC, dipalmitol lecithin, acts as a surfactant, enabling air to mix inside blood and preventing the collapse of the alveoli. Now, what happens in a respiratory disease condition? In a premature infant, due to immature lungs, dipalmitol lecithin do not synthesize enough. So there is not enough of dipalmitol lecithin, and due to which there is a collapse of alveoli of the lungs when there is breathing of air. Decreased oxygenation and blood and resultant in tachypnea, which is abnormal rapid breathing in infants. So that is how dipalmitol lecithin, which is a modified or a specific kind of phosphatidylcholine. So the correct answer is dipalmitol lecithin. Okay, there is a comment saying B is a component of lung surfactant, so that is why B is wrong. Okay, so B is not wrong, but the more specific correct answer is dipalmitol lecithin. So wherever, even if you search it or you read it online, you will find that DPPC, which is a modified version of phosphatidylcholine, is the correct answer. This is actually a phospholipid that is used as a surfactant. So this is the correct answer. If there would not have been dipalmitol lecithin, then phosphatidylcholine was the closest to the correct answer. If let's say it was phosphatidyl ethanolamine, then it would have been option B. Now that we have a particular modification, which is dipalmitol with the fatty acids belonging to the palmitic acid, then this answer is the correct answer. Okay. Is it clear to everyone? I just saw your thumbs up. Okay, great. Moving on to the 14th question. Mental retardation, blindness, muscular weakness, and accumulation of GM2 gangliosides are characteristics of which disease? Okay, so now a memory based question again. Mental retardation, blindness, muscular weakness, and accumulation of gangliosides are characteristics of which disease? Option A, Fabry's disease. Option B, metachromatic leukodystrophy. Option C, Tay-Sachs disease. Option D, Parkinson's disease. You have to read the question carefully. There are a certain set of symptoms mentioned here and the causative agent as well. The symptoms might be very similar, but you have to remember it via the causative. Why does it cause? What is the cause for this disease? Accumulation of GM2 gangliosides is the key to this answer. Which of these diseases has these symptoms as well as accumulation of GM2 gangliosides? Let me wait for some time and collect some answers. Okay, it's time. Let me take a look at the options. Aparna says C. Ramya is also saying C. Aishwarya is saying C. This condition mainly caused damage to nerve cells. Yes. So gangliosides, as we just saw earlier, so all these sphingolipids, ganglioside is a kind of sphingolipid, is abundant in the nervous system. So the correct answer to this question is option C, Tay-Sachs disease. Very good. All three of you have answered very well. Well done. So what is Tay-Sachs disease? Tay-Sachs disease happens due to mutation in the hexa gene. Hexa is a gene which can, when mutated, leads to beta hexosaminidase A deficiency. Now, due to this enzyme deficiency, there is an accumulation of GM2 gangliosides in neurons. So why so? So Tay-Sachs disease is actually an lysosomal accumulation storage disease disorder. Due to the deficiency of these enzymes, these ganglioside cannot be broken down and they accumulate inside nerve cells. Accumulation of these GM2 ganglioside in neurons leads to membranous bodies inside neurons, neuronal degradation and demyelination. So what happens in this Tay-Sachs disease? There is demyelination. The myelin sheath, which was so important in insulating the axons and in smooth conduction of nerve signals, now degrades because there is there is accumulation of these gangliosides. And there are independent membranous bodies forming inside the neurons. Cherry red spot in the macula of the eye and narrowing of the blood vessels. So these are some of these uh, diagnostic features of the infant or individual suffering from Tay-Sachs disease. And these are some other things to look at. Macrocephaly means enlarged head. Hypotony, mental and motor delay. So there is mental retardation, motor delay, difficulty in movement, convulsions, and this is the whole picture of Tay-Sachs disease, which happens due to an enzyme deficiency leading to accumulation of cerebral gangliosides, the GM2 gangliosides inside neurons. So in a normal neuron versus a Tay-Sachs disease neuron, accumulation of GM2 gangliosides leads to decreased exocytotic activity and GM2 accumulation were observed. So more accumulation of GM2 gangliosides lead to less firing of the neurons at the presynaptic terminal. Gangliosides are a type of glycosphingolipid. So these are not just sphingolipid, they also have sugars on them, found abundantly in the nervous system, particularly in the cell membranes of the neurons. So these are found in the cell membrane of the neurons. GM2 ganglioside is a specific ganglioside containing sialic acid and a unique carbohydrate structure. So this is a complex structure of sialic acid GM2 ganglioside. You do not have to remember it. This is just for reference. And what is the main roles they play? They play in nervous system: synapse function and stability, these gangliosides, cell recognition and communication in the brain, and cell membrane integrity. Now when these accumulation happen inside neurons, the membrane integrity is lost, myelin sheath is lost, and there are independent bodies forming in the cytosol of the neuron, the cell body of the neuron, like this you can see, which leads to decreased firing at the presynaptic terminal, which is then further leading to mental retardation and motor delays. Is it clear to everyone? Yes, very good. Cool. Moving on to the last question of this session. Question number 15. Waxes are esters of fatty acids with high molecular weight. So which of these waxes are esters of fatty acid with high molecular weight? Which of these could form? Option A, tetrahydric alcohols. Option B, trihydric alcohols. Option C, dihydric alcohols. Option D, monohydric alcohols. So which of these alcohol, when in reaction with a fatty acid, can give a high molecular weight, or is which is naturally found in waxes? You know, beeswax and paraffin wax, the candle wax that you see, these are all esters. Now which of these alcohols is commonly found in those waxes? The question is as such. I'll wait for a minute and collect your answers after looking at the chat box. Okay, let's not waste any more time and look at the chat box for some answers. Ramya is saying, sir, it's D. Maybe of being simple lipid. Aparna D. Aishwarya is also saying D. I want to tell you guys that the last question is also option D, monohydric alcohols. The answer to the last question, and you guys have answered very nicely. Well done. This session is going great. We have come to the last question, and you guys have answered all the questions, most of the questions correctly. Monohydric alcohols form high molecular weight esters with fatty acid, which are commonly found in waxes, in natural waxes. Now, what are waxes? Waxes are esters of fatty acid with long hydrocarbon chains and monohydric alcohols. They are hydrophobic and solid at room temperature. The candle wax is solid at room temperature. It is naturally found in plants, animals, and microorganisms. So, this is the reaction by which wax forms, known as esterification reaction, and the reverse of it is hydrolysis reaction. A carboxylic acid, which is a long chain fatty acid in our context, with an alcohol, which is the answer that you said, monohydric alcohols, in an esterification reaction will give removal of a water molecule, this OH, sorry, the H here and the OH here as a water molecule, and combination of this fatty acid with OR group, forming an ester. So these kind of esters are found in wax, normal waxes. Here is an example or a list of table of natural waxes. Fatty acid, the reaction RCOOH plus monohydric alcohol, R prime OH being a simple lipid. Wax RCOR prime plus H2O, where R is long alkyl chain from a fatty acid and R prime is a long alkyl chain from a monohydric alcohol, like a cetyl alcohol. Cetyl alcohol is found in beeswax. So beeswax has palmitic acid plus cetyl alcohol and used in cosmetics or polishes. Carnauba wax has longer chain fatty acids, and myricyl alcohol C30. You see how long these acids and alcohols are used in car wax and in cosmetics. Spermaceti wax, palmitic acid C16, and alcohol cetyl alcohol again from sperm whales. This was isolated from sperm whales, that's why it is named as spermaceti wax. So here is another schematic showing fatty acid plus a fatty alcohol giving a wax ester through removal of a water or de-esterification reaction, and the reverse reaction is the hydrolysis reaction. When you add water to an ester, then it will form a carboxylic acid and an alcohol. Is it clear to the students? This was an easy answer and you answered correctly. Great. So with this, we come to the end of our problem solving session. I'll close this slide and switch on. I'll stop presenting. I hope this session was informative and you could learn something from it. And we finished well before time. So all these lectures are recorded and this will be uploaded in the YouTube. You might be already having the link for the playlist. The slides that I have made will be shared with you via the folder, Google Drive folder link, and I'll be here to answer your questions, help you solve doubts. It will be a fun two-way session. We will meet every week. Okay, someone has raised a hand. Sir, where can we get these questions which you are presenting? Okay, so these questions are actually previous year's assignment question, which I am repurposing for you guys. The previous year and the current year do not match. Most of it does not match. That is why I was trying to make you solve these questions. So these questions are provided to me via the NPTEL to aid in functioning these assignments, right? So it might be a rare chance that you might get the exact questions in your assignments, but mostly it does not happen. The questions do not repeat. But be sure that these questions are from the same lecture series that you have been following in the NPTEL. So today we solved many questions from lipids, from Krebs cycle, studying, you know, mostly lipids and carbohydrates and sugars and DNAs. So all these has been covered in the fourth week lecture series. So the exact questions might not match in your assignments, and these questions are from the previous year's collective previous year's question from the same course, the basics in biology. Any other query you have, please ask. Otherwise, it's time, and I will end this session. See you next Saturday, where we will solve problem set for week 5. Read through your lectures and follow up with your quizzes and assignment. Don't miss the deadline. And I'm here for you for your live sessions. And whatever concept I just try so that all these students have clear concepts, because this is the only point of live classes that you will be having, right? Thank you for joining. I'll end this call. Bye bye. Take care.